Okay, so I'm going to talk a little bit about the EBI metagenomics portal, which is basically something developed by the EBI with the following objectives. So you will see that one of the first things that we want to do is actually provide the services to allow easy archiving of these data, so the sequence data and the metadata into the public archives. And we want to do that and encourage the submission of metadata, descriptive metadata that complies with minimum information standards. The reason that EBI thinks it's well placed to do this is because we've already got expertise in functional analysis of sequences, so we hope that we'd be able to provide robust services to the metagenomics community. And basically the essential idea is that we want to help laboratory researchers make sense of their data. It's as simple as that. The reason why EBI thinks it's well placed, as I said, is because we're basically a biological data hub. So we already have many different types of data, both nucleotide, protein, now even increasingly things like chemistry. So we're very used to handling it. We also understand, therefore, the challenges of doing large-scale sequence analysis and handling large amounts of data. We also being a hub, it means we can integrate with multiple different diverse data sets, and we have training facilities that many people, constantly in demand training facilities actually, where we can explain how people could use these resources and basically disseminate what we do to the world. So as you've already heard, metadata is really important if you want to give any sort of context to the analysis of what you've been sequencing. So, I mean, we've already heard, so it's the in-depth controlled description of the sample that your sequence is taken from in the case of metagenomics. So latitude, longitude, those are the simple ones, but obviously it could be as in detail as you wanted it to be. And the GSC minimum standards exist basically to help us structure these data and ensure that there's a minimum amount of information that's collected. And they do this often relying on controlled vocabularies and existing ontologies like ENVO. And essentially, the idea is that the richer the metadata, then the richer the analysis and the better the analysis you're going to have at the end. So this is why we really want to encourage the submission and use of metadata in the archives. So to submit metagenomic data to the archives via the EBI metagenomics, there's basically two ways you can do this. There's for small-scale users and for large-scale users. The small-scale users can use, we have a project ongoing at the moment with the ISA tools guys here at Oxford. So you can either use the ISA tools or you can use the native SRA webbing. And I'm just going to go through a little bit about how you would use both of those. They both have the same kind of underlying fundamental principle. So because they're for small-scale use, they're interactive. They use web or graphical user interfaces. You have experts creating a template which can then be shared with experimentalists. So they take the hard work out of sort of deciding what goes into the metadata collection. And these templates would then capture what minimum standards you want to have. So in the case of GSC, it would be compliance with mixed standards. And then the users can use these GUIs or other tools, like everybody knows that biologists tend to like using Excel, so there's a lot of spreadsheet interactivity to enter their data, and then they can upload it using a variety of formats and use it to submit to the public repositories. So now I'm going to struggle here because it's so tiny and I can't really read it here. So the first thing is so as I said, this is how ISA software suite, what's wrong? Oh yeah, but okay. I'm scared to move though because of the camera. So the ISA software suite is, like I said, one of the ways that people could enter data. We really want to give the options to people to be able to choose whichever tools they feel most comfortable with. So with ISA, there's an initial step, as I mentioned. So which one is it? Yeah, I've got it. So the configurator is used to create the templates. So a power user would create templates for then an experimentalist to to use when they're collecting their data. And then they would, the experimentalist is then able to use this template that's been produced, either using whatever spreadsheet. So this top bit is the spreadsheet that they're comfortable using. So Google Docs is given as an example here. Or they can use the ISA Creator tool to fill it in using this, using a specific tool produced by the ISA guys. And then this will produce a file. There's other tools as well that will produce this file format that can then be used to submit the data to the public repositories like the SRA hosted at EBI. But in the meantime, the SRA at EBI has also been producing a submission tool, an interactive submission tool. So they previously had the webbing tool, and they realized that it was important to support more checklist-related data inputs. So there's been a lot of effort put into the ENA version of a submission tool, and this is an interactive, as I said, interactive tool that can be run on the web. So you can upload your raw sequence data files using a special uploader tool. But then you can also select, and this has only been released for maybe a couple of weeks, so people probably aren't familiar with this. So there's, obviously SRA is a generic repository for DNA sequence data, so there are lots of different checklists. There's one for epigenomics. There's also GSC specific mix, so I don't know whether you can read, but there's like human associated, human gut associated, so there's been quite a lot of work to create these checklists, and Peter has been heavily involved, as well as Chris, who's also in the audience, in defining what the fields that should be in these checklists are. Once you've selected your checklist, you can then select which attributes you're going to upload, and you can also then fill in what default values you want them to have. So on the right here, I made up some data. This is my test thing. So my big fat metagenomic sequencing project. And then the idea is that if you've created this template in a similar way to the ICE tools, you can then add multiple samples that have the same sort of metadata captured and modify them depending on which specific attributes you've varied in your experiment. And it's not the only way that you can submit data to the archive, so you can also use the SRA RESTful web interface, web services interface. So again, if you're a larger scale user, you might want to use that instead of using the GUI. So those are the ways you would submit your data. We really want to encourage people to submit rich metadata. And one of the things that we've been discussing is internally is ranking the samples and the studies that have been sent to the archives according to the quality of the metadata. So we kind of thought of a three-tier way of thinking about things. So the first would be that you've got very high quality, it basically conforms to minimum standards metadata, but it's also metadata that's been curated, because obviously you can check for the presence of whether or not somebody's filled in a field, but whether they've filled that in with something sensible is another thing entirely. So we kind of want to distinguish between things that have conform to the minimum standards in the sense that they tick all the boxes, they've filled in all the fields, but also have good quality metadata in the sense that it makes there's biological sense there. And then the second is to have that, so this is sort of too high quality, which is basically it conforms to minimum standards, but one of them has been checked by a human other than the person who submitted it. And then low quality is that it just doesn't conform, and it wouldn't be highlighted in the interface in any way. And one thing that we're going to do is basically prioritize, because there's a lot of data in the archives already, we're going to prioritize the analysis of sequences that come from these higher quality, better described samples, basically, so that, because otherwise you can't really do much with them. It's important to have it well described. Just to give a bit of an overview of the type of analysis that we do on the samples. So essentially there's two main things that you want to do. You want to do diversity analysis of the taxonomy, but also functional analysis of what the communities in the samples are actually doing. We have a pipeline that's using a lot of quite standard, I would say, components to do predictions of RNAs, coding sequences, and what the functions are, and also to do taxonomic analysis. And then we display this on our web interface together with all the metadata that we've collected. So this, as you can see, is a marine sample. So you've got a little Google map. You can also then view the results of the analysis as it comes out of our pipeline. And we just basically give an overview of what's happened, what QC has gone on, what the predictions by interproscan and gene ontology are, and what we'll shortly be including the taxonomic predictions from the CHARM program. So I just really wanted to finish with saying why it's important to have metadata. Detailed metadata allows the construction of complex queries based on environmental context. So you might want to say, give me the alpha biodiversity for all samples found in saltwater. I mean, you can read that. So you wouldn't be able to do that without a decent amount of descriptive metadata. And if we're going to have true power, currently everyone's focused on their own individual projects. If you're going to have true power, it's going to be through meta-analysis of lots of different projects. So we really need to be able to utilize this metadata appropriately. So as far as EBI metagenomics goes, the future developments are that we're going to develop web services to allow simple access to this data. We're going to also develop a search engine to allow meta-analysis in this way. And also we're going to be further, we have to continuously improve our analysis pipelines and visualization tools. So these are our funders, our collaborators, special mention to the E&A team, Peter, the GSC of course, and the ISOTools team, and our competitors. And finally, I wanted to just say we're also recruiting. So if anybody knows of anyone who might be interested in working as a bioinformatician on this project, then it closes in a few days' time. Thanks.